Good morning, everyone on the West Coast. Good afternoon for everybody else. Welcome. My name is Greg Gisevich. Those of you who can see my video on here, I apologize in advance. They say the TV camera adds 10 years and 10 pounds. I think the Zoom camera adds 30 years and 50 pounds, but so be it. The Healthcare Laundry Accreditation Council is pleased to present this online webinar entitled How Monitoring Keeps Laundry Processes Effective, On Target, and Safe. Our speakers for this presentation are Lumay Gamboa, Chief Operating Officer of Bay Linen, Inc., Clearwater, Florida; Dane Brooks, Project Manager for Q Laboratories in Cincinnati, Ohio; Michael Lowenstein, Vice President of Quality and Scientific Consulting, Q Lab, Cincinnati, Ohio; and I'm Gregory Gisevich, President, CEO, Sterile Surgical Systems, Tumwater, Washington, and HLAC past president. Today's presentation will last approximately 60 to 70 minutes and includes a specific question and answer period at the end of the session. You can ask questions via the web conference system by simply typing your question into the Q&A box on your screen and then clicking send. We will be answering these questions at the end based on the questions that you submit. And please realize today's webinar is being recorded. All participant lines will be muted during the live program. Okay, so I will start here, and what you're going to learn today, and the audience, this is a really interesting audience that we have today. We have about 160 participants in the audience, and there's a pretty good breakdown between healthcare laundry people and people that work in a hospital. So we'll try to talk to both audiences. We realize hospital people might not be as conversant in laundry stuff, and laundry people might not be as conversant as in hospital things. So we'll try to speak to both audiences as well as we can. So we're going to talk about why monitor your laundry processes. What are some common contamination scenarios in a laundry? And then we're going to get behind some of the science behind these contamination scenarios. We're going to talk a little bit about what should I actually test in the laundry. Then we're very fortunate to have Lumay here, who's an accredited laundry, who's actually doing this process monitoring right now, so she can share their experiences. And then what presentation or meeting wouldn't be complete without your action items? So we actually have action items for you. And then finally, we'll leave about 10 minutes at the end for a Q&A period. So fundamentally, our job in the laundry industry and in the healthcare industry is to ensure that every piece of linen that touches a patient is free from contamination. And I think if we think about all the things in a hospital, we think of surfaces and we think of air and we think of all the things that touch a patient. Really, nothing's going to have a greater degree of contact with a patient than the actual linens on the gown or the bed or the towels or whatnot. And really, the kind of the good news in all this is outbreaks attributed to healthcare laundered textiles are pretty rare. So there have been 13 confirmed outbreaks in 49 years attributed to laundered healthcare textiles. That's a really, really good record. And there's a little bit of breakdown of where those were. And some of the pathogens identified in there. Laundry people, you probably can't pronounce these things, but infection preventionists, I'm sure you're well familiar with all of these. Even though those numbers look pretty good in our industry, it's not the whole story. So, there may very well be pathogens that have caused HAIs that we don't know about that originated from linen. So we know factually healthcare textiles can transmit pathogens. We also know that reports of microorganisms on healthcare textiles, they're actually pretty common. We know that multi-drug resistant microorganisms can survive for actually weeks on healthcare textiles. And I read most recently with COVID-19, I think COVID-19 lasted, it was like 4 days on linen, which is pretty long. And we also know that soiled healthcare textiles can contaminate surfaces and medical devices. So even if one of these things is the culprit in contaminating a patient, the soiled healthcare textile may have been the originator of that. HAIs in the U.S. in 2011 cost between 96 and 147 billion with a B. That's a really big number. That's a huge number. So this is an expensive problem, and it's 1 in 31 patients. That's 2015 data. It was 1 in 25 in 2011. So there is pretty good progress being made in this space. Hopefully some of that's attributable to laundry, but that's across the board in the hospitals now that we're measuring these numbers. And the types of HAIs occurring in U.S. acute care hospitals, this is 2011 data. This is the major sites of the infection. So there's pneumonia, gastrointestinal illness, urinary tract infections. These are the numbers for those. And I'm just going to give a couple examples here. So in New Orleans in 2009, there were actually tragically several deaths in children's hospitals, and this was traced directly to deadly fungal disease that was traced back to the linen. So lots of lessons there in that tragedy. And then more recently in 2015, there were 5 mucormycosis cases. Again, that's a fungus at a hospital in Hong Kong. So these things are, while rare, they're not unheard of, and they can be lethal in certain instances with immunocompromised patients. So what's our role in this? Well, this is pretty simple for us, right? We wash the linen and we dry the linen under recommended conditions, and everything's good. The stuff is clean and it's safe for patients, and everybody's happy. Is that right? Well, maybe it's not that simple. So what I'm going to do is I'm going to walk through some common contamination scenarios, and I'm actually going to go through a video of a healthcare laundry. Coincidentally, this is my healthcare laundry in Tumwater, Washington, and it's a short video, but what I'm going to do is I'm going to stop it in different areas to point out potential contamination areas. And what I want to say is, laundry people, you've probably seen this many times over. You know what the laundry process looks like. Hospital people, you've probably seen it too, but I'll go into a little bit more detail. This is kind of one way to do this. So here we go. Let's get started in the video. And right now what we're seeing is healthcare textiles on a conveyor ramp going up to feed a tunnel washer or a continuous batch washer. So what are some things that we notice right here? Well, the textiles are contaminated already, so we don't really worry about contamination here, but we worry about this guy's health and safety. He's wearing goggles, that's good. He's wearing PPE, that's good. He's wearing a hat, I think that's okay. This is an old video before COVID-19 time, so he would be wearing a face mask right now. One thing to add on here is these textiles have been soil sorted already. Very, very, very important is we want the textiles to be sorted appropriately so that we're using the correct wash formula on here for these textiles. So let's move on now. So the textiles are dropping into this continuous batch washer, and they're being washed right here. So are they getting clean right here? How do we know if these textiles are getting clean? Well, hopefully there's good wash chemistry that we can monitor the processes for that has enough chemistry, enough temperature, enough time, and enough water to make the textiles clean. So we're going to assume that these things are clean. Now we've extracted them. This device here, it's called an extraction press. Many continuous batch washers have them. So these are clean textiles coming out of here. Or are they clean? So we monitored our processes in our plant in Tumwater, and I might add, we have the most compliant healthcare laundry in the entire planet Earth. I'm being a little bit facetious there because guess what we found? We found this water actually was contaminated. And we found this surface right here actually was contaminated. So based on monitoring these processes, we actually made a change. We changed the cleaning chemical that we used here to a sporicidal cleaner. We changed the frequency and the method for doing that, and we also changed the same things for that surface. So that's a kind of a process monitoring precursor for later in the presentation. So let's move on. Now it's here. Now it's going up into a shuttle that's going to go into a dryer. Now, is this air clean? If not, guess what? We've contaminated this linen. Is this surface clean? Guess what? If it is not clean, now all of a sudden, this linen is contaminated. Now we're going into the dryer, and now we're discharging from the clean. Surface clean, is this surface clean? So now we're going into an ironer. Are the people's hands feeding? Now we're going into a picker. Again, we have more hands, we have more air. Now we are going into an ironer, and again, we want, are these surfaces clean? Is this air clean? Here's a pillowcase folder. Are all these devices contaminated, or are they clean? There's more hands, there's more surfaces, there's more conveyors, there's more air again. These are just some scrubs going in. Again, we have these same contamination guys that are in there. More hands. Now we have bodies that are actually touching them. More machines touching them. Going up the scrubs, and that's the end of our process. And however, is it really the end of the process? And so here's the patient or the linen being used by the patient in the hospital. Even though this is a talk about laundry processes, one thing that I just want to add, we need to keep this stuff clean, not only in the laundry, we need to keep it clean on the trucks, we need to keep it clean on the actual linen room in the hospital, in the linen closets, and actually in the linen room or the patient room of the patient for this little guy here to be safe and for all this stuff to be clean for him. So fundamentally, the question that we're asking is, how do we prove what we do in the laundry to validate to the hospitals that our linen processing meets their requirements? And the answer to that question is process monitoring. So I've given kind of a high level of the laundry process and some scenarios there. I'm not a science expert, so let's dig a little bit deeper, and I'm going to turn it over to Michael, who is a science expert, and he's going to give some of the science behind process monitoring.
Hello, everybody. Good morning, good afternoon, good evening, wherever you are in the world. As Greg was saying, there's all sorts of sources of microcontamination, and I think it's helpful to consider your laundry as a manufacturing process. The clean linen is your finished product, either that you're making or that you're purchasing if you're a hospital person. So there's many sources of microcontamination. Diagram at the bottom, there's a version of that that's used in most industries, whether it's the food industry, the drug industry, specialty chemicals. It's just a way to think about what are all of the inputs to my process that could be a source of microcontamination. And as Greg was saying, it's one thing to make something clean, but then it's another thing to keep it clean throughout the process. So it's important to remember that microorganisms are ubiquitous. That means they're present everywhere at once. They're in the air, they're on surfaces, they're on people, obviously, they're in water, they're everywhere. And what's more, laundry facilities are not designed to be sterile. So there's not any special design controls to keep microorganisms, to keep you free of microorganisms. You may have processes in place to keep the microorganisms level low. That's by design, but how do we make sure that that's being effective? There is essentially no environment where microorganisms cannot exist. So I know it might be tempting to think that, well, I've got a whole bunch of chemicals in my laundry process. Nothing can survive that. It's a dangerous thing to think. Oh, I'm putting it through a really hot drying process. Nothing can survive that. That's a dangerous thing to think. Much better to think of the air, the water, the surfaces, your people as ingredients into your finished product, all of which could be contributing microcontamination to your finished product, the clean linen, and want to have control over all of those processes so that you can feel confident that you're maintaining high microbial integrity throughout. Some people might think, well, I'll test the finished product. I'll test my clean linen at the end to show that it's free from microcontamination. That certainly can be part of your process and is probably a good thing to do, but there is no product testing scheme on the planet that is going to be statistically significant, right? You laundry folks could tell me, I'm not sure how many pieces of laundry you're cleaning at any given time, but it's a lot. So in a very scientific term, we know it's a lot. You can't test it all. So it's really important to make sure that all of your processes are monitored, all of your sources of microcontamination are being monitored to make sure that they're staying in control, not sterile, but they're staying in control. And then you can be sure that you have a system of high microintegrity. So you monitor the process to give confidence that your finished product is going to be safe for the end consumer. Greg, you can go to the next slide. Thank you. So as I said, the goal here is not to be sterile, but it's to maintain a system or systems to keep an environment of high microbial integrity. But how do I make sure that the procedures that I put in place are effective? Well, I should monitor those systems and look for microcontamination and see if it's what I expect to see. So like I said, one of the main ingredients, if you want to think of it as such, is your environmental air. Laundry is like all manufacturing environments. I'm sure you guys have cardboard. You've got the laundry itself, right? These generate lots of airborne particles which carry microorganisms around. So we want to monitor the bioburden in the air to see, is it in control? Is it normal? Is it what I expect to see? Or is it increasing over time? Is there something that might indicate that I might have an issue? Same thing with the surface. I think Greg pointed out, hey, I've just cleaned my laundry, but if the surface I put it onto is not clean, then I'm going to recontaminate my laundry. Water. We use water to clean the laundry, but water itself is one of the worst harborers of microcontamination. So we want to make sure that our water, we're not in a way that's going to increase the bioburden over time. And then, of course, our people are an ingredient in our process. If they are not following good hand washing, they're also going to contaminate the laundry. Those are sort of our systems that we'd want to monitor over time. And then we've got our finished product testing. That's the testing of our textiles at the end to make sure that they are free from hazardous microcontamination. So QLabs can do all of this testing for you. You can get all of this testing done, and you can do it once if you want, but that's a very limited value because these processes are dynamic, right? You guys are cleaning different things all of the time. People are coming in and out. You're changing formulas. All sorts of things are changing. So the value is to collect these data on some frequency. That way you can track and trend over time. What you would recommend is to determine what is statistically normal for your facility, and then you can set alert and action levels to say, if I go beyond this range in one of my systems, I know there's probably something wrong with that system. It can target your investigation and target your corrective actions in the event that there are any issues. And
so, Mike, I'm going to interject here a little bit, and just to kind of echo what you said, which is right on. If we test one washcloth in a laundry that processes 50 million pounds per year, what does that tell us? Kind of doesn't tell us anything. It tells us that one washcloth, but I mean, I think if we have a regular system of testing this washcloth and other things regularly, we can get a lot more intelligence and actionable intelligence from that data.
You got it. And I think the example that you gave is, hey, we know what the normal bioburden on this particular surface is, but then, we test that every month, say, for 6 months, and then in month 7 we test it, and the level is twice as high as it was before. We know that maybe we should go take a look at that facility, or that portion of the facility. Has something changed that might explain that? And collecting these data and trending them over time puts you in a proactive position rather than having to react to every contamination event of the finished product. If you're monitoring the process the whole time, then you can proactively address potential issues before they have a negative impact on your finished product.
Right on, Mike. Thank you.
Yeah. And of course, designing the program is what HLAC is helping to do and what QLabs can help consult with. So if there's any questions about how to design a program, we'd be happy to consult with that. So I've talked a little bit about what you should test, why you should test it, and I'm going to pass it over to my colleague, Dane Brooks, to talk about how he'll conduct these tests for you.
Thanks, Mike. Hi, everyone. I'm Dane. I'm Q Laboratories Research and Development Project Manager. As Mike mentioned, he went through all of the ways that you can test, so I'm going to go through the 5 segments of the kit that we put together. That's textiles, air, water, surfaces, and hands. Before we move forward with that, I do want to talk to you guys about a few definitions. You'll be hearing me use some words that you may not be familiar with while I'm explaining each piece of the kit. I want to quickly cover what these terms mean. One is microbes. These are germs, bacteria, bioburden. For our purposes, consider all these synonyms. 2 is sampling. This is the way we are capturing a portion of the microbial load in a facility to be analyzed in the lab. And the 3rd is auger plate. This is germ food, a way we are going to grow microbes so that we can see them and report how many there are. Mike, you can go forward with the next slide. Sorry, Greg, you can go forward with the next slide. There we go.
I knew you meant me.
Yep, I did. The first test we will cover is textile testing. It's simple to collect. You just place the textile in the bag, and when it gets to the lab, we will analyze it for bacteria, yeast, mold, and anaerobic spore formers. We'll use QLabs techniques that give accurate measurement. The common industry practice here is to stamp the fabric with a Rodak plate. Rodak plate's intended use are for hard, non-porous surface samplings. So Rodak plates do not penetrate the fibers of the textile. The technique is a good technique, but used for this application, it'll yield a low microbial sampling potential. We use a liquid extraction method and pulverization to penetrate the fibers of the linen. The extraction yields higher microbial loads and better represents the bioburden throughout the textile.
So, Dane, I'm going to interrupt you. If you
Sure.
do a Rodak plate test on, let's say, a pillowcase, and you come up with, 15 CFUs per square decimeter, this is a completely unfair question, but what's using your method of bioburden recovery, what can we expect to see?
Yeah, so typically if you're using a liquid extraction with pulverization, your results are going to be logarithmically higher than a Rodak plate. So say you would see 15, you may see something more like a 150 or even a 1500 count if you're getting into the webbing of a fabric. You really are leaving quite a bit behind if you don't get all the way through the matrix of the fabric and extract all the bioburden within that.
And the reason I bring that up is because we did this testing, and we always, with the Rodak plate tests, were under 20, and then when we did this method, we were in the hundreds, and I nearly died when I saw that, but it makes complete sense based on what you just said. You're recovering more bioburden than, because you're getting all the fibers, not just that surface area stuff.
Right, and textiles in a plant can vary quite greatly. You know, your webbing can be very thick. It can be made out of different materials. Higher levels of cotton tend to hold higher levels of bioburden, so you might have a clean surface, but you know, if you have a thicker webbing, it might be deeper into the fabric. It's where you're carrying up quite a bit of bacteria that can come to the surface later on through its use.
Right.
If you could go to the next slide. Okay, the next test we include in the kit is air testing. Mike spoke about the importance of monitoring your facility's air quality. I want to take you through a couple of ways to sample air. There's passive or active. Both have their advantages and disadvantages. To passively sample, an auger plate is placed in a sampling location for a given time, and microbes falling in the ambient air are collected on the plate. The advantage of this technique is that it's easy to perform. You just lay down the plate, start a timer, and it does not require any specialized equipment. The disadvantage is that there's no way to measure the volume of passed over the auger plate. The advantage of active sampling is that a calibrated volume of air is passed over an auger plate. This decreases the variability in the test and allows for more compatible data generation, collection, and collection. The disadvantage is that it requires specialized equipment, but we felt when we were putting together the kit that it was important to include that piece of equipment so that the facilities could compare their data from collection to collection.
So I'm going to interject here again, and the question I'm going to ask is, where would we want to do this air sampling in our laundry?
Yeah, absolutely. So that's up to each facility to decide the location they want to sample, but anywhere you have a critical zone, that is what we would recommend. So somewhere where folding is going on, where there's a lot of air traffic, a lot of airflow is somewhere where you would want to sample, and commonly it's done in the folding areas.
And also, I mean, I guess I would think dryer entry or dryer exit, or any folding area, or anywhere where clean textiles are exposed to air.
Right, which is the whole facility, right?
Well, no, it's the whole clean facility. So basically anything out of the washer, anything into the washer, you can be as dirty as you want as long as your people are protected.
Absolutely. Pre-wash, you really don't have to, that's more about PPE and transferring your PPE whenever you go into the clean side, making your hands, but once you get to the clean side, right, that's where you would want to sample.
Again, yeah, think of air as an ingredient. You know, it's an ingredient that matters after you've gotten your laundry clean. So that's where the air as an ingredient matters. Is we've cleaned up our laundry. Now we need to make sure we keep it clean. Air is an ingredient that we can't keep out of the laundry. It's going to be exposed. We want to test the air. And as Dane said, you don't want to go stick this off in the corner where there's no airflow, where there's no people walking, where you can probably be pretty sure that the micro load is going to be low. You want to put this where it matters, where your clean laundry is exposed, in an area of high traffic, in an area of high airflow.
Yeah, absolutely. So let's cover surface testing. So we provide two materials within the kit, ATP swabs and Rodak plates. And previously in the PowerPoint, I mentioned that Rodak plates are not great for textiles, but they are great for surface sampling. Rodak plates provide a direct measure of cells on a surface. The downside to these plates is that the results aren't available until they're incubated for a few days, so you have a delay on when you can get your results, and that's why we include the ATP swabs as well. I do want to mention a few common areas that people do use RODAC plates. They're conveyor systems, carts, and folding stands. Both of these are post-wash, post-dry, so you can see your surfaces in that area are all clean. The ATP swabs, they measure adenosine thiophosphate, not cells. ATP is the energy currency of cells, and ATP can be detected in dead and non-viable cells. So we include the ATP reader so you can take a quick swab, get a reading, and you can take action immediately in that area. But ATP is not as reliable as the RODAC plates, and that's why we include the RODAC plates as well, so you can have a little bit better data to trend over time. But if you find a dirty area with the ATP swabs, you can clean it right away.
And then following up on my other question, what are some good surface areas? And Lume, this one I'm going to direct to you. What are some good surface areas that we would want to measure for their surface contamination?
We test working tables or conveyor belts where the linen is transferred out of the equipment directly onto bulk carts.
Okay, great. And I'll answer my own question. So we test conveyors, we test work surfaces, we test clean linen carts to verify that they're actually in fact clean, and feeder portions of machinery we test. Okay, I'm moving on, Dane.
Sure. So next we'll do water testing. Water testing is of course critical to the industry, and a lot of the facilities probably are already doing water testing. One of the differences that we're including with our kit is that our sample cups include a little bit of sodium thiosulfate powder. And sodium thiosulfate is a neutralizer that stops the antimicrobial activity of chlorine. What this does is it prevents the microbes from dying to the chlorine during the shipment of the samples to us. So once you've sampled your water, you can get an accurate snapshot of what the microbial load is at that time. You don't have to worry about it steadily dying during shipping, which will happen if you don't have sodium thiosulfate included in your sample cups. Okay, the final test that we conduct is the hand hygiene testing. As we've seen in previous photos, some laundry processes involve manual or by hand preparation. So what we've found over time is that there's a high variability in hand washing effectiveness from individual to individual. So we developed a patented method to recover both the pre and post hand washing efficacy, so you can measure how well your employees are washing their hands. This method also checks underneath the nails, a frequently forgotten area during hand washing. And our last slide for me here, I just want to summarize everything that we've covered and kind of tell you a little bit more about the kit itself. We've covered a lot of different microbiological principles, and it may seem daunting to perform testing. You've got 5 different aspects of your facility to test, but it's not that difficult. The toolkit is designed to come with everything you need to do all of these tests. Each segment is color-coded to prevent confusion, and there are easy-to-read instructions for each segment. There are videos that walk you through how to do the testing, and QLabs is always available to provide live customer service if you do get stuck. And that's everything I wanted to cover here, Greg, so if you want to move on to Lumi.
Okay, so I'm going to segue it this way. Thank you, Dane. Thank you, Michael. That was super informative, and thanks for trying to sell us on this process monitoring toolkit, and especially thank you for selling us on how simple it is. But I don't believe you, because you guys are trying to sell this thing. So that's why we brought Lume in here, who's actually used it, and she can confirm or deny everything you said. So we're very lucky to have Lume with us here, who's been doing this every month for about the past year. So she is the expert on actually using this, what her experience has been, what she's learned, and what she's changed with it. Take it away, Lume.
Thank you, Greg, and hello, everyone. I'd like to start by sharing a little of who we are and why we have chosen to participate in the process monitoring program. Bay Linen is a co-op laundry in Clearwater, Florida, currently processing 50 million pounds annually, which includes over 60,000 sterilized towel packs and over 120 gown packs with 2 in-house autoclaves. We service the Baycare Health System in the Tampa area, Sarasota Memorial Hospital, and Lee Health System in Fort Myers. Overall, we service 22 major hospitals and many more of their offsite locations, totaling 6,100 hospital beds. That's a significant number of lives we feel we impact, and we are proud to do so safely. Bay Linen chose to participate in the program in response to our customers' needs for more information on our processes and a means for us to have the data to monitor the outcome of these same processes and be proactive at the same time to changes that may be necessary to continue meeting their expectations. Annually, we are inspected by infection prevention nurses and a quality committee that meets 6 times a year, all who now rely on Bay Linen's participation in this program, as well as our HLAC accreditation. For us, the process monitoring kit is a validation in the entire process of turning the sole received into safe, hospital-grade, acceptable linen. Just like we perform biological testing on every load sterilized, this is how we go about ensuring the rest of the linen is also acceptable. Next slide, please, Greg. Next, I'm going to tell you a little bit about how we conduct our testing. Together with our member hospitals, we chose to conduct monthly testing. This we felt was going to give us the most amount of information. Once the agreement was shared with Q Labs, the kit arrives timely and always ready for use. On the day of testing, we split the testing supplies between 2 team members. An engineer handles the textile, air, and water samples, and a quality supervisor handles the hand hygiene and surface testing. For us, the textile testing consists of 3 items, like a pillowcase or a towel and a baby blanket. We consider these items high-touch patient items. Air sampling is conducted in 3 different departments, like sterile processing, clean side, and specialty, which handles all their scrubwear, dietary, hypoallergenic, hyperbaric, and pediatric linen. Water samples are collected from 3 sources: the main supply line, the cold water tank feeding our tunnels, and all 3 tunnel discharges. Surface testing is conducted in different working surfaces, like we shared before. We test stainless steel work tables as well as rubber conveyor belts. Hand hygiene testing is done on different team members, representative of different departments throughout our facility, as we make sure our hand washing procedure is implemented well throughout our facility. Overall, the kit here at Bay Linen takes an estimated 6 hours to complete, from getting the supplies ready, to conducting the testing, to completing the test forms that are returned to Q Labs. A week after, we received the reports from Q Labs, and we have been very pleased with their response time to answer any questions or schedule time to review results. Next slide.
So, thank you, Louis. I'm going to interrupt you for a minute. One of the things that people are really afraid of is how challenging is it to do these tests, and what was the ramp-up period for your people that are doing it? So.
Not very long. The kit comes with a complete instruction on 1 page for every of the tests we conduct, and it's very user-friendly, all color-coded. It always comes to you in the same, packaged in the same order. It's not very difficult at all. We picked up right away.
Great, thank you.
On our last slide, I'm going to share a little bit of what we've learned throughout this year. We've learned to set alert and alarm limits, which we feel enhance the data review process by taking it to the next level. And what I mean by that is the limits are set to prompt different activities, from reviewing wash formulas used for items exceeding limits to initiating additional cleaning to retraining team members. Since using the kit, we have implemented weekly hand hygiene testing and training as our data became variable for some time and we wanted to see it level. As we all know, hand hygiene is universally viewed as one of the most critical factors in infection prevention, especially nowadays. We feel all our training has proven effective with COVID-19. We have been able to keep our workforce healthy and safe, along with the correct use of PPE and other policies. So making sure policies are effective and implemented works. Surface testing has reassured us our processes are working and current EVS team members are reliable in using them. We have also updated the cleaning procedure for all clean side conveyor belts, including the frequency and the cleaner disinfectant we use to do so. For us here, the entire program has become an essential part of keeping us very engaged in the review of all our existing processes and procedures. We believe the data being collected through this process monitoring program is crucial to what is going on in the world today in our industry, as we reassure our customers our commitment to deliver hygienically clean linen that is safe for their patients.
That's great, Lumay. Thank you so much. So My Laundry has also done this program, although not as frequently as Lumay's Laundry, and I would like to share another story, and I wish there was a poll here because I could ask questions. This is kind of like a test question for you all. So we had a situation where we tested linen out of the dryer, and the linen was clean, the linen was acceptable, and we tested the linen on the table, the finishing table specifically, and it was contaminated, or had much higher level of contamination. So we tried to trace this into what it was, and I wish I could ask people, but I'll ask our panelists. Can anybody guess what the problem was that caused the contamination? So the problem was, very interestingly, it was an employee handling it, not with his hands, but with his body. So it was someone reaching into a tub, grabbing a bunch of washcloths, and then rubbing them, on his body, and his clothes were contaminated. So that was the source of that. And I bring that example up because one of the great uses of this tool, not only doing all the statistical stuff and doing the regular benchmark, I think that's great, but we found it to be a great debugging tool. So a great way to zoom in on exactly, okay, this was clean in this area, this was contaminated in this area, how did this happen? Oh, we found we were able to zoom in right away. Without that, it would be much more challenging to do that. And we all know all kinds of contamination dangers lurk, hidden ones all over the place in a laundry, and this is just a great tool to help us find those and mitigate those. So thank you, Lumay. That was super useful and real world, which we always like. So I'm going to now talk about your action items in this, with this entire process monitoring and the process monitoring toolkit. So laundries, monitor your processes. It's going to make your laundry better and safer. And what do I mean by that? Hopefully you're accredited. Hopefully you're following all the standards, the 640 odd standards to show that you're doing things right, but monitor them. So you have a checklist to clean your carts. That's great. Verify that you are actually cleaning them. You have a checklist for hand hygiene. You have training for hand hygiene, like Lumay is doing. Actually verify that that's happening statistically, and these things are going to make your laundry better and safer all along. Hospitals, specifically infection preventionists and EVS people and materials people, hold your laundry accountable. Make sure not only they're getting accredited, but make sure they're monitoring their processes. Ask to see some of the results of them. It's going to make your linen safer for your patients, and it's going to provide critical, actionable data to the laundry. And then finally, laundries and hospitals, the more people that do this, the more process monitoring data it's going to equal more data, which is going to equal better benchmarking, which is going to equal safer laundries and ultimately safer patients. So what am I talking about here? Well, I'm always asked, and I think Mike and Dane are always asked as well, what is good and what is bad? The first time I did this, I got my results back, I almost fell off of my chair because I thought, oh my good heavens, these counts are massive compared to what we've seen with the Rodak plate linen tests and other testing that we've done. What are we doing wrong? And it turns out that we're probably doing a lot of things wrong, but in this particular case, this great toolkit and this great technology is finding everything. We call it the Hubble telescope of laundries because you have a granularity of data that wasn't available before, and that's super, super, super useful stuff. But the more data we get, we can start doing now statistical analysis. When you give your data into QLabs, it's all confidential. So it's between, you're the only one with access to the data, so we're not going to publish that. But what we are going to do is we're going to collect that data, anonymous data, and actually do some statistical analysis on it. So wouldn't it be great to see something like, here's the average bioburden load of clean healthcare textiles. Here's the average of contaminated ones. Wouldn't it be even better to go deeper? Here's the average for cotton. Here's the average for polycotton. Here's the average for all poly. Or better yet, here's the numbers for different seasons. Or better yet, here's the numbers for different geographies. This is all super, super useful stuff for us as an industry, and I think it'll help us all get better and ultimately result in safer patients. I'm going to ask Mike and Dane, can you dig a little bit deeper on how to set up a program in this? You know, what are you, Mike, you were saying you can't test everything, so tell us more. How do you, what's a good way to use, and other good ways to use this to make your laundry better and learn actionable data?
Sure. So, and I think you spoke to this a bit in the beginning, is, you know, we know the laundry when it, or the textiles, when they're coming into the laundry, we know they're dirty by definition, right? Doesn't do a whole lot of good to monitor the process there, because the laundry is contaminated already. It's going to be cleaned. We don't need to worry too much about it there. It's really after it's gone through the laundry, and I know each laundry process is slightly different, so you need to look at your process and decide, okay, at what point do I think my laundry materials are clean, you know, whether that's after, you know, the initial wash, whether it's after water extraction, whether it's after your dryer, or if it's after all of those stages. You know, after the first clean, I think my laundry is clean. Now the rest of it is just processing steps. Then you'd want to, as I think as Dane correctly called them out, those are critical control steps then, right? I've taken the time to make it clean. Now I need to keep it clean, and so I put in procedures. And I have, you know, if I'm an accredited laundry, I'm going to have, you know, not just design controls, but I'm going to have procedures to have clean carts, I think is the one you mentioned. How do I ensure that those are all being effective? Well, that's where I want to put in a monitoring step. So, for example, if I'm thinking about monitoring air, I don't need to monitor the air probably at the very beginning of the process, because the laundry is dirty and I know that any air that's going to be coming in contact with the laundry is not going to negatively impact it. But after the dryer, I'm done with cleaning the laundry. It's dry, it's ready to go be folded and sent out. I need to start worrying about the air recontaminating it now. So I might want to monitor there. Same thing with surface monitoring, right? Up in the beginning, if it's a dirty laundry coming in contact with the surface, I don't need to worry too much about the surface getting it dirtier. But after I've cleaned it, if I've cleaned my laundry, I want to make sure I'm putting it on a clean surface. How do I make sure that my surface is clean? Surface monitoring. So you just want to think through your process, and it might be slightly different for different facilities. It really depends on how your facility is laid out, how you move the laundry through your facility. Because if your dirty laundry and the clean laundry, if they share the same room and they share the same air, well, then maybe you do want to monitor your air earlier in the process than another facility that maybe that's in a separate room. So it really depends, but it's common sense, right?
You just spoke healthcare laundry blasphemy. So, yeah, but it's
Good.
okay. And we obviously, like, we never want clean linen and dirty linen in the same area. But if we were to do that, which we would never do, this would be a great way to monitor that.
Well, I guess when I meant same room, I meant is the HVAC system, are they totally separate, right? Do they have different supplies and different extractions? If they share the same air at all, if there's any pass through, if air doesn't care if a door threshold, that helps.
Right.
But the air could be moving throughout. So if you're in a facility where you don't have, they might be in separate rooms, but they're not on totally different ends of the facility, say, if you might be concerned that there's some air passing back and forth.
Right.
And you might want to investigate that. But anyway, like I was saying, the real point is, once I've cleaned the laundry, I want to keep it clean. So that's where you would want to monitor your process the most.
And we have an old adage in the laundry industry: it's easy to get the linen clean, it's challenging to keep the linen clean. And as we know, that's the journey. So I am going to thank you very much, Mike. I'm going to segue in. We have time for some questions here, so I'll go into the Q&A portion of this right now, and I will answer some, and I will also hand some out to our panelists. So the first question I have is, can you elaborate a little bit more? Hey, this is Blaze Bass. Hey, Blaze, how are you doing? Can you elaborate a little bit on how soiled linen is segregated inside the facility once it is unloaded off of the trucks? How do you ensure that the environment is kept sanitary and staff are safe? What does the space where the segregation is done look like? Lu Mei, do you want to take a crack at that, or would you like me to?
Well, then for us, as you said previously, the two must, it is sanitary and it should be kept some clean, but the soil linen should always be separate from the clean, right? And the team members working there should all be donning the appropriate PPE, correct?
Yes, yep, perfect. And then we want to make sure that soiled area is completely separated, and there's no opportunity for that soiled linen to touch clean linen, or even that soiled air to touch clean air. So great question. Okay, should we be sampling the mops and rags used to clean around the hospital as well? Absolutely, yes. So if it's going to be used to clean the hospital, and we want to ensure that those are clean, so this is a great tool to use to measure the bioburden load on those mops and rags. Another same question. That's good. Which area is recommended to surface test? So we've answered this, but let me just reiterate. So any surfaces where clean linen is going to touch are good surfaces to test. So that includes your clean carts, your folding tables, your conveyors, your entrances to machines, your exits to machines. In a hospital, clean linen rooms, clean linen carts, clean linen closets, and actually linen rooms. What is the minimum maximum amount of contamination, CFU per area or weight of textile, that can be measured? And we don't have that number right now, and that was why we have that action item to please give us more, please do this more so that we can have some statistical information on that. But there aren't absolute pass-fails for this yet. Lots of people have asked for that, but I think this more has to be within your facility and looking at trends and looking for outliers and establishing your own baselines based on your testing. Did I get that right, Mike?
Yeah, if that was the question, is there a specification? As you said, the answer is no, right? This industry is a little bit in its infancy. I can compare a little bit to other industries that QLabs does business with. It takes a number of years and a lot of people participating in this kind of self-regulating activity to determine, hey, what's normal for the industry? What's a level that we can all hold hands on and agree we should be below this threshold? In the meantime, all you can do is compare to yourself over time to say, you know, this is statistically normal for me, and I now know I've deviated from my statistical norm. I know there's probably a problem.
Great. Okay,
yeah,
next question.
Greg, if
yeah, sure.
the side of that question is what is the minimum and maximum amount of contamination that can be measured, that question. So we have 5 different tests, and if you're talking about the concentration of microbes that we can see here in the lab, it really varies test to test, but I see that they mention here textiles. For our textile test, we go down to a less than 20 value, and I see another question here compares that to the TRSA program. You may know that the TRSA program, their maximum is 20, but as I talked about in those slides, because we're using a different extraction technique, we're finding a lot more microbes. So our LOD is 20, and we can go up into the thousands, 10 to the 4th, 10 to the 5th, as far as what we can measure inside of a textile. The portion sizes that we collect are 5 grams for that test as well. And now each of the tests is a little bit different. The LOD on the water test is 1. The LOD on the hands test is 5.
Sorry, what's an LOD, Dane, for our
sorry. LOD is the limit of detection.
Okay, thank you. Everyone I know knew that, but just for the one person that maybe didn't.
Right. Then for the surfaces, that one's a little bit unique. We can really go up about as high as possible, except for on, you know, as high as what you'll ever see in a facility. We can go to 10 to the 6th, 10 to the 7th, which is millions and tens of millions of microbes. But for the air sampling and the Rodak plates, we can reliably count 10 to the 2, which is hundreds, but anything past that, there are too many microbes for us to really distinguish, and we can provide an estimation if it's at 10 to the 3 thousands. But anything past that, it's really hard to determine how many are actually there. And that's for the surface testing and the air testing, but all the rest.
It's kind of who cares, right? At those high, really high counts.
Right. If it's that high, you know, it's pretty dirty.
You're doing something wrong.
Right.
Okay. So moving on, what is the cost of the kit and the testing? So I don't want to give the cost exactly right now because honestly I don't know it offhand, but I do know that it's on the HLAC PMTK website and there are volume discounts. So if you do it once a year, it costs so much, and if you do it twice a year, it's so much, and all the way down to once a month. I can't recall exactly what that is offhand, but we will, when we do the write-up on the slides, by the way, these slides will be available. We'll answer these questions so that will be out there. Okay, Another one, making data available is inevitable, and are there companies that are developing tools or devices to enable this? What is your view on data monitoring becoming formally regulated and monitored? So I think this question deals with, hopefully I'm interpreting it correctly, is will this ever become mandated? And I can't answer that question from a federal law perspective. Will laundries ever become regulated like the FDA regulates sterile pack rooms? I don't know the answer to that. I do think, though, the better we do as an industry, the more of this type of stuff that we do as an industry, the more we can use the cliche kind of police ourselves, and as an industry, the less likelihood there is in the United States anyway that this will be federally regulated on that. How's that for a wishy-washy answer for everybody? Okay, another question. We have in our dirty income incoming cloth, an extractor with a filter that eliminates all microbacteria. When the cloth is removed, it works. So I think, let me try to interpret the question. Hopefully I'm getting it correctly. I think in the dirty room, there's a filter maybe that's the filter is filtering things out, and then when the cloth is removed, how's the air? What I would say to this question, again, hopefully I'm interpreting it right, is I think that's a great opportunity to actually test your air with some real data, to see how your air is doing with the filter and to see how your air is doing without the filter, and you can get some actionable data based on that. Another question, what is the criteria for success, the minimum and maximum of the current method? Again, I don't think we have not yet established those numbers yet. Am I missing any questions, guys? Nope, that's not one. I'm facing difficulty. I don't see any additional questions. Does anyone else, any panelists, do you see any additional questions?
No, Greg.
Okay, so with that being said, wait, did one just come in? I will say thank you for participating in today's webinar, How Monitoring Keeps Laundry Processes Effective, On Target, and Safe, brought to you by the Healthcare Laundry Accreditation Council. Special thanks to our speakers, Lumay Gamboa, Dane Brooks, and Michael Lowenstein. Immediately after this webinar, you will receive a survey. Please take a moment to provide us your feedback. In the next few days, you'll receive a link to a recording of the webinar. If you have additional questions or comments, please contact the Healthcare Laundry Accreditation Council at info@hlac.org. Today's webinar is copyrighted by the Healthcare Laundry Accreditation Council with all rights reserved. This concludes today's program. Thank you. You may now disconnect.